Good morning and Assalamu alaikum, class 9. Today we will carry on the chapter number 4, cells and tissues, which is the first chapter of your new class. So in the previous lecture we discussed a lot about the microscope, the types of microscope, and the magnification power of microscope, the resolution of microscope, the definition of the cells, the discovery of microscope. In today's class, we will discuss the differences between the electron microscope and the light microscope. So the comparison between light and electron microscope are that the radiation type of the light microscope is light, while the radiation type of the electron microscope is beams of electrons. The lenses used in the light microscope are the optical lenses, while the lenses used in the electron microscope are magnetic lenses. Magnification power of the light microscope is 10,000 times greater than the naked eye, while the magnification power of the electron microscope that it is 100 times greater than light microscope. The resolution power of the light microscope is 500 times of the naked eye, while the resolution power of the electron microscope is 400 times of the light microscope. In the light microscope, we can see two-dimensional images, which are also known as 2D images, while in the electron microscope, the transmission electron microscope shows the two-dimensional images, while the scanning electron microscope shows three-dimensional images. If you remember in the previous lectures when we discussed the types of electron microscope, so we discussed in the detail the transmission electron microscope give us the two-dimensional images. Just they give us the image internally, while the scanning electron microscope give us the three-dimensional images from all the sides. So these are the differences between the light microscope and the electron microscope. Now, after the discovery of microscope and microscope were discovered, it was becoming easy for the scientists to study the cells in detail, the structure of the cells in detail, the tissues in the detail. So, after the discovery of microscope, some scientists do the developments and they proposed a theory which is known as cell theory. So, how the emergence of the cell theory takes place with continuous developments in microscopy, cells of organisms were studied in detail. Here are the some steps that resulted in the emergence of theory called the cell theory. So, different scientists contribute a lot in the field of the microscope and the cells in the tissues, the structure of the cells, the discovery of the cells, and thus a new theory was proposed by the scientists, which is known as the cell theory. So, the first of all, I'm going to detail or to discuss. I'm going in the detail or to discuss the contribution of the Robert Hooke in the discovery of the cell theory. First of all, the Robert Hooke was an English scientist who discovered cell in 1665. So, cell was discovered by Robert Hooke. Please, students, highlight these for your information because MCQs repeatedly they are there in the paper in MCQs. So, please highlight them. Who discovered cell in 1665? So, who discovered the cell in 1665? Robert Hooke discovered the cell in 1665. He observed piece of cork under his self-made microscope. You can see in the diagram that he studied the piece of a cork, his self microscope. So he discovered the cell for the first time in 1665, and he was studying the cork, the cells of the cork under the microscope, his own made microscope. He observed small chambers like honeycomb structures and he named it cell. The next contribution was of the Anton van Leeuwenhoek. He was a Dutch scientist. He studied a drop of water under his own microscope. If you remember, I told you that Anton van Leeuwenhoek make a much better microscope. His magnification power was very high. So he studied a drop of water under his own microscope. The magnification power of his microscope was 300x, he noticed tiny creatures swimming in the drop of pond water. He was the first man to observe single celled organisms called unicellular organisms. So, first of all, the unicellular organisms were discovered by or studied by the Anton Leeuwenhoek in his own microscope. Next scientist who contributed was the Jean-Baptiste de Lamarck. In 1809, the Jean-Baptiste de Lamarck, a French scientist, also observed cells when he examined the parts of animals and plants under microscope. He concluded that all living organisms are made up of cellular tissues. Clear? Jean-Baptiste told that all the living organisms are made up of cells and tissues when he studied the living organisms under microscope. The next was scientist who contributed in the cell theory was the Robert Brown. In 1831, a British botanist, Robert Brown, nucleus found the nucleus in the plant cell. He discovered the nucleus in the plant cell. The cell was discovered by the Robert Hooke. Remember, the cell was discovered by the Robert Hooke, while the nucleus was observed by the Robert Brown in 1831. Next scientist is the Matthias Schleiden. In 1838, a German botanist, Matthias Schleiden, studied plant tissues and made the first statement of the cell theory. He stated that all plants are made up of cells. That Matthias Schleiden in 1838 studied the plant cells and he told that all the plants are made up of cells. Next scientist is Theodor Schwann. In 1839, a German zoologist, the Matthias Schleiden was a botanist, while the Theodor Schwann was a zoologist. Schwann found that all the animals are made from cells. As the Matthias Schleiden gave the statement that all the plants are made up of cells, he was a botanist, while the Theodor Schwann was a zoologist, and he also gave the concept that all animals are made up from cells. The Schleiden and the Schwann proposed the cell theory. So, in its Italian form, all living organisms are made up of cells. The next scientist who contributed was Jan Evangelista Perkinje. He was an English scientist. In 1840, he proposed that all the cellular containants are living materials and give them the name protoplasm. Jan Evangelista gave the cell the name protoplasm. And what is protoplasm? The living part of the cell is known as protoplasm. The next one scientist was the Rudolf Virchow. In 1855, the Rudolf Virchow, a German physician, gave the hypothesis that every cell comes from pre-existing cells. Omnicellular acellular. It means omnicellular acellular. Rudolf Virchow gave the statement that all the living organisms are made from the cells from प्री एग्जिस्टिंग सेल्स मतलब पहले से वजूद रखने वाले सेल से नए सेल्स बनते हैं. The Louis Pasteur, the most popular uh, bacteriologist who is known as the bacteriologist Louis Pasteur. In 1860, the French chemist Louis Pasteur, scientist Louis Pasteur experimentally proved the hypothesis of Rudolf Virchow. He experimented on bacteria and found that bacteria are produced from pre-existing cells. So, the Louis Pasteur was a bacteriologist and he gave the statement that all bacteria are produced from the pre-existing bacteria. These are the different scientists who work in the formation of or the formation of the cell theory. The first one scientist is the Robert Hooke who discovered the cell. The second one is the Matthias Schleiden who proposed the cell theory, who formulated the cell theory. He gave the main points of the cell theory. The next scientist is the Theodor Schwann. He is also the proposed the points of the cell theory collectively. The Theodor Schwann and the Matthias Schleiden proposed the cell theory. The next one scientist, the Rudolf Virchow, who gave the name protoplasm to the cell. So these are the different scientists which contribute in the cell theory. So in today's lecture, we discuss the history of the emergence of the cell theory, that how cell theory was proposed by different scientists. The main contributors are the Robert Hooke, who discovered the cell, the Anton Leeuwenhoek, who discovered the microscope, the Jean Baptiste de Lamarck, who examined the unicellular plants and animals in the cell, that all living organisms are made up of cell. Robert Brown discovered the nucleus in the cell. The Matthew Schleiden, a botanist, discover the cell in the plant. Cedar Sean discover the cell in the animals. Jan and Glista, Rudolf Firk and Louis Pasteur give their different